Hey there, and welcome back to our discussion on the cardiovascular system. Part one of our discussion on chapter 19 was kind of a short one, and if you will recall, my rationale was essentially I wanted you to take some time to make sure that, as this slide indicates, you understand the direction of blood flow as it makes its way throughout the circulation. So, what I encouraged you to do was using this slide as a study guide, make sure that you basically have it down pat, always knowing where blood is going to go next. So you want to always know that after blood is in the right atrium, it goes to the right ventricle next, then to the pulmonary arteries, to the lungs, pulmonary vein, back to the left atrium, and so on and so forth. So if you are watching this video, that must mean that you feel pretty comfortable with that by now. So that's great. So let's go ahead and continue. So our next discussion is going to center around, now that you have a firm understanding of where blood is going to go next, understanding, of course, blood is circulated throughout the body. Now that we know where to expect blood to go next, we want to understand, why does it go there? Why does blood flow in one direction and not the other? So this is going to be one of the most fundamental principles that we will discuss in this chapter and upcoming chapters. Blood flow is driven by changes in pressure, or rather differences in pressure between two different locations. So to get you to hopefully understand this, I want you to imagine something for me. So imagine that you have a balloon that is filled with air. So it doesn't have to be a real big balloon. It doesn't have to necessarily be inflated a whole lot. Just imagine that you have a regular old latex balloon that is filled with air. So imagine that you fill this balloon up with air and then you let all the air out through the open end. So you untie the little open end there and you let the air come out. So when you do that and you kind of pinch off the end there and then you let it go, all that air is going to come rushing out all at once. So why is it that the air did that? Why did the air just so rapidly move from inside the balloon to outside the balloon? So the answer to this question is that fluids meaning both liquids and gases, and it's going to be really important to understand this because this will apply to both liquids and gases moving forward. All fluids will naturally move or flow from areas of high pressure to areas of low pressure. So if air exited the balloon, then that must mean the air pressure inside the balloon must have been higher than what we would call the atmospheric pressure, the air pressure that is all around us. So all the air needed in order to move from one location to another was the opening that was formed by you opening the little end on the end of the balloon. And that allowed the air to move from high pressure to low pressure. So since we're talking about blood is a liquid, so therefore it is a fluid, and it is going to behave according to this principle. Now, what your heart is going to do, and we'll learn an awful lot more about the heart as we go through here, what your heart is essentially doing is acting as a gigantic pressure generator, essentially a pump. So what your heart is doing is squeezing the blood that is in different compartments or atria and ventricles of the heart. It is squeezing the blood and raising the pressure of that blood, so the blood will then flow from that area of high pressure to an area of lower pressure. So what the heart essentially does is pressurize the blood to ensure that it is going to move to a location that has a lower pressure. And by doing that, we ensure that blood circulates throughout the body, always moving from high pressure to lower pressure. So these pressure differences, which exist all throughout the circulation, starting with the heart, with the right side of the heart, going to the lungs, left side of the heart, out through the aorta and into the systemic circuit, these pressure gradients or pressure differences exist all throughout the circulation, and they are what are responsible for the continuous and constant circulation of the blood. So to demonstrate this, look at this graph right here. So this graph on the y-axis is showing us the mean systemic blood pressure. So at any given point in the circulation, whether it's the aorta, the arteries, the arterioles, the capillaries, the venules, the veins, or the vena cava, we're keeping track of the average pressure on the blood in that location. Now, if you start with the aorta here up top and you move from left to right, so from aorta to arteries and so on, you will notice that that corresponds to the correct direction of blood flow from the aorta to the arteries, arterioles, capillaries, venules, veins, vena cava, and now we're back to the right side of the heart, back to the right atrium. So if you look at the pressure graph here, you will notice that the pressure is highest at the aorta, and then as it moves through that correct flow of circulation, the pressure continues to get lower and lower. So the fact that blood moves from the aorta to the arteries is because the pressure in the aorta was higher than the pressure in the arteries, so high to low pressure. Blood moves from high to low pressure. The fact that blood moves from the arteries into arterioles is because the pressure in the arteries is higher than the pressure in the arterioles. The fact that blood moves from the arterioles to the capillaries is because, again, we're sounding like a broken record at this point, the pressure in the arterioles is higher than the pressure in the capillaries. So as blood moves throughout the circulation, it is continuously moving from high pressure to lower pressure, that pressure to an even lower pressure, that pressure to an even lower pressure. And it all culminates in the lowest pressure location in the body, which is the vena cava, which is our entry point back into the right side of the heart. So because the vena cava is the last thoroughfare to return deoxygenated and nutrient-poor blood back to the right side of the heart, you want it to have the lowest pressure in the circulation because you're ensuring that all blood is eventually going to make its way back there. And once that blood gets back to the right atrium, we can pressurize that blood again and start the whole process over once again. So if we take another look at kind of the internal anatomy of the heart, and we haven't really talked about a whole bunch of this outside of talking about the 4 different chambers of the heart, if we take a little bit of a closer inspection of this, I think a lot of what we're going to see here is going to make more sense now that we kind of understand what all the pressure is all about. So what many of these structures that we're going to look at here are designed to do is to ensure that that blood is always moving in the correct direction. We want to make sure that blood in the right atrium only goes to the right ventricle and not backwards through the vena cava. Any blood that is in the left ventricle, we want to make sure goes through the aorta, not backwards into the left atrium. So keep that in mind. So we understand that anytime blood moves from one location to the next, it's moving from high to low pressure, high to low pressure, high to low pressure. But when blood is in the heart, as opposed to when it's in kind of the general circulation, whether it's pulmonary or systemic, the heart presents some unique challenges for us. For example, there will be times during one cardiac cycle in which the pressure on the blood in the ventricles, meaning the right ventricle here and the left ventricle here, there will be times when the pressure on that blood in the ventricles is actually higher than the pressure in the atria, meaning the right atrium here and the left atrium here. So that causes a little bit of a conundrum for us, because if the pressure in the right ventricle here is higher than the pressure in the right atrium here, that would dictate, based on what we said, that blood in the ventricle must go backwards back into the atrium because that's moving from high pressure to low pressure. But if that were to occur, blood would be moving in the incorrect direction. So we want to have certain fail-safes in the heart to make sure that this does not occur. So essentially what we're asking here is, what about the heart, what structures within the heart prevent this backflow or regurgitation from happening? We want to make sure that even when the pressure in the ventricles is higher than the pressure in the atria, we don't want blood to go that way. So another way of kind of phrasing this question is, how is it going to be guaranteed for us that blood will flow in the correct direction from right atrium to right ventricle and left atrium to left ventricle? What is preventing the backflow of blood there? So what we are going to see here is the 4 different chambers of the heart contain a set of heart valves. Little doors, little fleshy connective tissue doors that are going to open and close, open and close, open and close, to make sure that this backflow or regurgitation of blood does not become an issue for us. So these heart valves come in pairs, and you will find a pair between the atria and the ventricles, and a pair between the ventricles and the arteries. So let's start with the heart valves that separate the atria from the ventricles. Those are called AV valves, or atrioventricular valves. You have one on the right side of the heart, and you have one on the left side of the heart. The one that separates the right atrium from the right ventricle is called the tricuspid valve. The one on the left side of the heart that separates the left atrium from the left ventricle is called the mitral or bicuspid valve. You can hear it called either one. For the sake of consistency, I'm only going to call it the mitral valve. So the idea here is that once blood flows from the atrium to the ventricle, regardless of which side of the heart we're talking about, when blood moves from the right atrium to the right ventricle or left atrium to the left ventricle, it must have done so because the pressure at that time was higher in the atrium than it was in the ventricle, correct? Okay, so after that delivery of blood occurs, then something is going to happen that will cause the pressure in the ventricles to be higher than the pressure in the atria. So, like we said, the problem that creates is that would lead you to believe that the blood would move backwards back into the atria, but we don't want that to happen, and our AV valves here are going to prevent that. So these valves are going to, like I said, open and close, and they do that in response to changes in pressure between the atrium and the ventricle. So the rule of thumb here is that your AV valves, which regulate the passage of blood between the atrium and the ventricle, these guys will be open when the pressure in the atrium is higher than the pressure in the ventricle. So think about that for a second. If the pressure in the atrium is higher than the pressure in the ventricle, this would lead you to believe that blood will flow from the atrium to the ventricle. Well, that's exactly the way we want blood to flow. That is correct. So in order to allow that flow of blood to occur, anytime the pressure in the atrium is higher than the pressure in the ventricle, we want our AV valves to be open because that promotes the correct flow of blood. But like I said, in situations when the pressure in the ventricle is actually higher than the pressure in the atrium, that would promote the regurgitation of blood backwards, and we don't want that. So as soon as the pressure in the ventricle gets higher than the pressure in the atrium, that is when your AV valves snap shut. And when they snap shut, even though the pressure gradient would dictate that the blood moves backwards into the atrium, because we close off that pathway, blood can no longer move that way. And that now highly pressurized blood in the ventricle only has one other way to go, and that is out into the arteries, which again is what we want. Okay, so with that being said, let's look at our other pair of heart valves. These valves, called semilunar valves, are going to separate each ventricle from their corresponding arteries. So for the right ventricle, the semilunar valve there, the pulmonary semilunar valve, separates the right ventricle from the pulmonary arteries, and then the left ventricle is separated from the aorta by the aortic semilunar valve on the left side. So even though these valves are called something else, they are going to function almost exactly the same way. They are going to open and close in response to pressure differences between the ventricles and the arteries. So the semilunar valves, more specifically, will open in response to ventricular contraction. So when that cardiac muscle around the ventricles contracts and squeezes the blood to pressurize it, that increase in pressure is going to be what forces those semilunar valves open and allows that blood to be ejected out into either the pulmonary circuit or the systemic circuit. So similar to what we did with the AV valves, we can come up with a set of binary rules for when the semilunar valves are open and closed. So for the semilunar valves, when the pressure in the ventricles is higher than the pressure in the arteries, that's going to be when your semilunar valves are open. And again, that is promoting the correct flow of blood from ventricle to artery, ventricle to artery. So in those cases, we want the semilunar valves to be open. But once a full ejection of blood has occurred and the pressure in the ventricles goes back down below the pressure in the arteries, that's going to be when we want those semilunar valves to snap shut to prevent any arterial blood from coming back through the aorta or through the pulmonary artery and back into the ventricles. We do not want that regurgitation of blood, so we use the semilunar valves here, opening and closing. Opening to allow blood to go in the direction it's supposed to go, closing to prevent it from coming back the way it's not supposed to go. So another very interesting thing to talk about here, just on the basis of how these valves are structured and kind of how they work to open and close to either allow blood flow through or to shut blood flow off completely. Other than not having heart valves at all, another possible way that kind of things could go wrong for us here is if one of those heart valves prolapses on us. So essentially what we mean here, I'm trying to get my hands in the right place here. So if this is one of your valves right here, so this is what it looks like when it's open, and then this is what it looks like when it's shut. So open, shut, open, shut. So another way that this could potentially go wrong is if we go from open to shut, but a prolapse would mean that those little flaps are going to go down too much, meaning that even though we're trying to shut it off, we still have an opening for blood to go through. So we do not want our heart valves to prolapse. So that would mean that it closes too far. It goes way over the edge in closing, and those flaps are going to kind of stick up into the atrium, which is what we do not want. So for this reason, we have attached to the base of the ventricles little strings of connective tissue, which are sometimes called your heartstrings. These are called Chordae Tendineae. So essentially what they do, the way I like to think about it, the piece of imagery I like to use is think of a drawbridge. So you don't really see drawbridges much anymore, but if you kind of think of like some of that old medieval imagery of like a castle with a moat around it and a drawbridge to allow passage across the moat, then the drawbridge is going to have ropes attached to it. So you can pull on the ropes to bring the drawbridge up. You can use the ropes to lower the drawbridge down. That's not unlike what the Chordae Tendineae are doing. But more specifically, what these Chordae Tendineae do is they place tension on the bottom parts of these mitral valve and tricuspid valve flaps, meaning that when they close, that tension increases, ensuring that we can't close the valve more than what is permissible. So essentially what these Chordae Tendineae are there to do is to prevent any prolapse of either the tricuspid or the mitral valve. Okay, so now that we have not only a good appreciation for the direction of blood flow, how pressure is involved in getting blood to flow, and the role of the heart valves in making sure that blood flows through the heart in the correct direction, let's talk a little bit about heart sounds. So in a clinical scenario, one of the most commonly performed tests or procedures is that either the doctor, the nurse, or some other practitioner is going to take a stethoscope and move the stethoscope around different places around the heart to listen for different sounds. Now, of course, we understand that the heart makes sounds, but what is it exactly that is producing those sounds? Well, this is a perfect segue since we just got done talking about our heart valves. What is producing those audible sounds that you hear when you listen to someone's heart is the closing of those AV and semilunar valves. It turns out when those valves close, they produce an audible sound, a sound that you can pick up on if you're listening with a stethoscope. So AV valves close, that produces a sound. Semilunar valves close, that produces a sound. And because, as we are going to become very acquainted with later on when we talk about the cardiac cycle, because the AV valves close first, and then shortly after the semilunar valves close, one cardiac cycle is going to produce two heart sounds. So bum bum, bum bum, bum bum, bum bum. AV valves close, semilunar valves close. So these sounds through onomatopoeia are verbalized as lub-dub, the first heart sound corresponding to the closing of the AV valves, and the second heart sound corresponding to the closing of the semilunar valves. So what these sounds correspond to, and we'll become more acquainted with this later on, so if you're not quite on board with what I'm about to say just yet, it's not a big deal, but definitely make sure that you're on board with it by the time that you finish part 3 of this video series. So the sounds, the first heart sound and the second heart sound, correspond to ventricular systole. So systole refers to the period of time in which a certain part of the heart is going through contractions. Usually, most of the time when we just generally say systole, we mean ventricular systole. So we mean the ventricles are contracting, that cardiac muscle is contracting to pressurize the blood and eject blood out through the arteries. So you will notice here all the way at the top in graph A here, you can see these dotted lines that are provided to tell you when systole is and when diastole is, diastole referring to a period of relaxation. You can see that from the time the first heart sound starts to the time that the second heart sound starts, this is the period of ventricular systole. So systole lasts from the time that the AV valves close to the time that the semilunar valves close. So that's going to be something that is very, very convenient for you to remember going forward. Like I said, if you're not quite there with it just yet, that's not a big deal, but definitely make sure that you feel comfortable with it later on in the chapter. So for someone with a normally functioning heart with perfectly healthy heart valves, A here, this graph at the very top, is what your heart kind of audiogram should look like. So you should see a fairly short first heart sound and a fairly short second heart sound. So the sound is produced by the vibrations of the blood as the heart valves move through the blood to go from an open to a closing position. If these hearts, so this picture right here just indicates that the first heart sound, the lub, is the AV valves closing, both the left and the right going at the same time, and then the second heart sound is the dub, which is the semilunar valves closing. So, like I said, graph A here is what a normal heart sound audiograph is supposed to look like. If any of those sounds look abnormal, or sound abnormal, for any particular reason, whether you have extra unexpected heart sounds or elongated or abnormal heart sounds, meaning the sound is either louder than you expect or the sound lasts for longer than you expect, these are what are called heart murmurs, and they indicate to us that we have an abnormal flow of blood through the heart. So just to give you a couple of examples here, look at graph B here. So what you'll notice here is that we have a very, very loud and very long first heart sound, while the second heart sound looks fairly normal. So because the problem here is with the first heart sound, this almost absolutely has to tell us the problem is with one of the AV valves. There's some problem with the AV valve. Either it's not closing all the way and it's causing it to kind of flap in the blood flow, meaning it produces an extra long sound. Based on the fact that the abnormal heart sound or the murmur is the first heart sound, that tells us that the problem has to be with the first or AV heart valve. So again, with graph C here, you see not exactly a very loud murmur, but you see definitely an elongated first heart sound. Again, that probably tells us that the problem is with the AV valves, which is appropriate since this is called mitral regurgitation. There's a problem with the mitral valve, one of those AV valves, meaning essentially it's not closing all the way, and as blood kind of regurgitates back into the atria, that tissue kind of flaps in the wind like that, or flaps in the blood, which produces that kind of extra elongated sound. For D here, you can see the first heart sound actually looks pretty normal, but this time it's the second heart sound that looks kind of weird. This is aortic regurgitation, meaning that similarly there's a problem with the aortic semilunar valve. So kind of your takeaway from this should be, when you're listening to someone's heart, you can get or infer a lot of information based on which heart sound sounds weird. If the first heart sound sounds weird, it's a problem with the AV valve. If the second heart sound sounds weird, it's going to be a problem with the semilunar valves. Okay, so before we really start talking about kind of the major function of the heart, which is getting blood into the pulmonary circuit so that we can oxygenate that blood, and then pushing blood from the left side of the heart out into the systemic circuit so that we can nourish all those different organs and tissues like the brain and the liver and the kidneys and all that. Before we start talking about what the heart does for the rest of the body, let's talk a little bit about how the heart nourishes itself. You got to take care of yourself before you can take care of others, right? So if you look at the aorta here, so the aorta, you can see just the very top of the aortic arch here, and then you can see how the aorta branches into a number of different other major arteries, which are going to feed different places throughout kind of the upper half of the body. The bottom or descending arch of the aorta would then service everything kind of below the midline of the heart. So one of those outlets that comes off of the aorta that is visible right here, right at the base of the aorta, about on your screen, about an inch below the aortic arch. You can see it labeled as the left coronary artery. The aorta, before it really gets into that arch, will have a quick little outlet which branches into the coronary arteries. And these are the arteries that are going to provide the cardiac muscle of your heart with all the oxygen and nourishment that it needs. So we've talked back in chapter 10 about how energy consuming muscle contraction is, and since your heart is literally beating 60 to 80 times a minute for every hour, 24 hours a day for your whole life, it's burning through a lot of energy all the time. So we absolutely have to make sure that we are getting a constant supply of oxygen and nutrients to the cardiac muscle cells of the heart. So when you hear about coronary arteries, you probably most know about this in the context of when things go horribly, horribly wrong, right? So we're talking about heart attacks or myocardial infarctions. So a infarction refers to the blockage of an artery into a particular tissue that cuts off the flow of blood to that particular tissue or organ. If you have a myocardial infarction, meaning an infarction of the coronary arteries, it's a heart attack. If you have an infarction in the carotid artery or other major arteries that feed into the brain, that's going to produce a stroke. So it's the same sort of problem, but it's going to produce different results based on which artery was blocked. So most commonly, the cause of this is through atherosclerosis, which is something that we'll talk about later. So atherosclerosis, which is the accumulation of these fatty plaques in the artery that narrows the space that blood can flow through and can cut off blood flow. Again, if this atherosclerosis occurs in the coronary arteries, the end result most likely is going to be a myocardial infarction if it is not remedied fast enough. So here you can actually see an angiogram in which someone has had a contrast dye injected into their bloodstream, and you can actually see through the dispersal and diffusion of this dye throughout the bloodstream, you can pick up on where blood flow is not good, because you can see with this red arrow right here, you can see that blood flow through this left coronary artery here is not good. It indicates that there's some type of blockage there. And then you can also see a blockage of a circumflex artery right here. So you can see kind of that narrowing right there, right where that arrow is. So that tells us that there's probably something obstructing blood flow there. More than likely, it's some sort of atherosclerotic plaque. But other than that, you can see all the branches of these coronary arteries feeding into arterioles and, of course, capillary beds. And from kind of the pathway that these different vessels take, you can kind of see the overall silhouette of the entire heart here, and you can hopefully appreciate how all of those arteries and arterioles are servicing the cardiac muscle of the various myocardia of the heart. So when you have atherosclerosis of coronary arteries, that is going to produce a lack of oxygen in those cardiomyocytes. Anytime this happens to a particular tissue, in which case they run out of oxygen or they lack a sufficient amount of oxygen, this is a condition called ischemia. So Most commonly, this is going to happen in the brain or the heart when you're talking about really, really, really bad ischemia. It does happen pretty commonly in skeletal muscle tissue when you're working out, but that's not a big deal. That just produces the kind of muscle soreness and fatigue that we talked about in Chapter 10. But for reasons that we're going to talk about, this is a much, much, much bigger deal for the heart. So like I said, ischemia of the heart or of the brain will happen usually as a result of atherosclerosis narrowing the diameter of an artery and preventing the natural and good flow of blood and delivery of oxygen and nutrients. So the consequence here is very similar to what we talked about with skeletal muscle. With skeletal muscle, if your muscle fibers run out of oxygen due to overuse and overstimulation, they're going to have to switch over from cellular respiration, which produces a lot of ATP, to glycolysis and lactate fermentation, which does not produce a lot of ATP. That wasn't that big a deal for skeletal muscle because it is adapted to being without oxygen. It just means it's going to get tired very easily. So cardiomyocytes are going to be a little bit different. So cardiomyocytes are going to rely heavily on lipids for their energy. They can't really rely too much on carbohydrates, glucose, or a stored up supply of glycogen. So we don't really have that available to us like we had with skeletal muscle. And cardiomyocytes are desperately, desperately reliant on aerobic respiration. Essentially what that means is that cardiac muscle absolutely cannot tolerate being without oxygen, hardly for any time at all. So if you do have atherosclerosis of any of those coronary arteries, that cuts off blood supply and produces ischemia in the cardiac muscle, those cardiomyocytes almost immediately are going to start dying by a process called necrosis. So the longer your cardiac muscle is without oxygen, the more and more of that tissue is going to die, and that is the myocardial infarction. The end result of a myocardial infarction is that a lot of that cardiac muscle tissue is going to die off, and its function is never, ever, ever going to come back. Something that's also kind of worth mentioning, not so much because it's that important to us right now, but it's something that has only really relatively recently been shown, and it's something that you may have or maybe will hear of in the future: ischemia reperfusion injury. So let's say that you have a patient that comes into your ER or your hospital or wherever you are that has suffered a heart attack, and your immediate inclination is probably, okay, they have a blockage in a coronary artery, we need to restore blood flow right away. Well, not so fast. Let's hold up on that just a bit. So ischemia, like we said, refers to a lack of oxygen in a particular tissue. So that is a huge problem, but at the same time, you want to be careful about suddenly flooding the affected tissue with all the oxygen that they could desire, meaning you want to be careful about how you go about restoring blood flow. Ischemia reperfusion injury is what occurs to cells that have been ischemic for a while, and now all of a sudden we reperfuse them, meaning we reintroduce full blood flow, and now suddenly they are going to be introduced to high levels of oxygen that they maybe are not quite so used to on the short term. So it's thought that cells are kind of slow to adapt to kind of things getting back to normal so fast, so you actually may have even more cells die as a consequence. So that's something that's kind of odd, kind of worth mentioning there. And then the last thing to really say about myocardial infarctions for the time being, and I kind of referenced this earlier, one of the big reasons why heart attacks are such a big problem is not because there's a good chance that if you have a myocardial infarction, you might not survive it, but even if you do survive, your heart is never, ever, ever going to be the same. Dead heart tissue cannot be replaced because cardiomyocytes are post-mitotic. This is something that we kind of referenced way back in Chapter 3. We said that certain tissues, in fact, most cells in the body, are post-mitotic, or what we also call quiescent. These cells, like cardiomyocytes or neurons, they are quiescent, meaning that they have permanently exited from the cell cycle. So if you do have a heart attack and you do lose a lot of cardiomyocytes, the remaining surviving cardiomyocytes cannot simply just divide to replace what has been lost. It's not the same as if you get a cut on your skin and you heal it up by using cell division to replace the cells that were lost. There are no stem cells in the heart that are capable of regenerating tissue. But it's for this reason that stem cell therapy is such an interesting conversation to have, because the idea there is that if we could get some type of multipotent or pluripotent stem cell that is capable of generating healthy functioning cardiomyocytes, we could potentially plant those into the heart of someone who has had a heart attack and possibly get those to heal up and replace those cardiomyocytes that were lost. So one thing to make sure that we understand, just to kind of make sure we're on the same page here, hematopoietic stem cells, what we talked about in chapter 18, the type of stem cells that you find in red bone marrow that generate new red blood cells, white blood cells, platelets, etc. Even though the heart pumps blood, cardiomyocytes and these blood cells are not at all related. They are not at all in the same developmental lineage. So it's a nice thought, but unfortunately, cardiomyocytes cannot be replaced with hematopoiesis, just to make sure that we understand that. Okay, so next what we are going to get into, and really what we're going to kind of finish this discussion out on, although we still have a ways to go on this, we need to talk about the electrical conductance through the heart. So we understand, of course, from the end of chapter 10, that cardiac muscle tissue, when it contracts and produces its own action potentials, it derives its depolarization from pacemaker cells. So what we want to talk about here is we want to understand how, from the time those pacemaker cells produce their pacemaker potentials and we get that depolarization spreading through gap junctions through the cardiac muscle tissue, we want to know how does that produce the contraction of the atria followed by the contraction of the ventricles, which is how it's supposed to be. So again, we've talked about how the heart beats on its own because of these pacemaker cells and how the sympathetic and parasympathetic divisions of the ANS are both capable of either speeding up your heart rate or slowing it down. But the ANS is not at all capable of stopping your heart or starting it back up at all. So a couple of things to remember from chapter 10. Don't forget, depolarizing stimuli will spread from cardiac cell to neighboring cardiac cell through the gap junctions that are present in intercalated discs. And don't forget that we call those units of contractile units that are all connected by gap junctions, we call those units myocardia. One myocardium contracts as one unit separate from other myocardia. So, as we've discussed before, the heart will continue to beat after you sever nervous innervation to it. It's totally different than what we saw with skeletal muscle. So if you sever the nerves that feed into skeletal muscle, if you sever the nerves that contain the axons of those somatic motor neurons, you have paralyzed those skeletal muscles. They will never contract again, at least not consciously. The heart is different. If you sever sympathetic and parasympathetic stimulation to the heart, it will continue to beat and contract on its own because, like we said, it derives its depolarization not from the nervous system, but from those innate and intrinsic pacemaker cells that are a part of the heart itself. So to help demonstrate this, I have this video for you to watch here. So the slides are already posted, so you can watch this video on your own, but let's go ahead and take a look at it here.